Okay, let's get back to the presentation. So I'm the only one talking about infectious bronchitis virus in this conference. So because of that, I thought to give a slight background. And I'm going to talk about the molecular characterization of the virus. As Dr. Kawaja indicated about the precision vaccination, characterizing the field virus is really important. So I'm going to talk about the molecular characterization. Then I'm going to talk about some pathogenicity studies, particularly the impact of the virus on the egg production. When the virus infects the birds at the age of 26 to 30 weeks at the peak of production, and also when the birds are infected as early as 1 to 14 days. And also I'm going to talk about the immune suppression caused by the IBV. This is not very new. We know about chicken infectious anemia virus, we know about Marek's disease virus, and the IBV, those are the immunosuppressive virus. But I can say that IBV has got some immunosuppressive properties, but those information need to be confirmed. And also I'm going to talk about the vaccination. These are conventional vaccines, and I'm going to talk about the vaccination program and how it can mitigate these pathogenic features. So the IBV is a coronavirus. It's a positive sense, single-stranded RNA virus. It is enveloped. That's a good thing, so that it is vulnerable to any detergent, any disinfectant. And also I want to show a couple of, actually the 4 structural proteins which are very important. One is the spike protein and the membrane protein, or the M, then the nuclear protein, or N, and the viral genome. The avian coronavirus belongs to the genus gamma coronavirus, which is quite distant from beta coronavirus, where the SARS coronavirus belongs, MERS coronavirus belongs. When I see this slide, it is really scary. See, every single coronavirus originated in bats. Then it jumped to several species and ended up in the humans as well as domestic animals. If you focus on the bovine coronavirus, you can see that it has jumped to humans, it has jumped to canines, pigs, and also the equines. So it looks like coronavirus can jump to any animal species. Why? And what is the basis? It is because of the RNA genome, and that is vulnerable to the mutation, and also the recombination. The way the coronaviruses replicate lead to the recombination very easily. IBV exists in dozens of serotypes, then genotypes, lineages, and all those put together, there are hundreds of variants of this virus. This is really a challenging situation. One of the viruses I'm going to talk about today is called Delmava 1639 virus, which popped up in Canada somewhere around 2017, and it's a continuous problem. And these genotypes and the strains, they can induce specific diseases. It is a respiratory virus, as the name suggests, but it can infect the renal system, gastrointestinal system, and the reproductive system. There's one strain that replicates in the muscles. That is the 491 strain. I heard 491 is present in Pakistan. Delmava 1639 virus is very similar to the QX strain, which is also present in Pakistan. So until 2017, in Canada, 491 was dominant. It was causing problems. And at that time, I can remember I was the newly recruited faculty member, and the government of Canada, who are responsible for registering vaccines, they consulted me to ask, should we import 491 live attenuated vaccine? Since I was thoroughly knowing about the nature of the coronaviruses at that time, I said it is not advisable. If there is a inactivated vaccine, yes, but live attenuated vaccines are going to create more and more problems because of the recombination and also for the mutations. I was thinking, what if I am wrong about the 491 vaccine? Fortunately, after a couple of years, 491 disappeared. And the DMV 1639 came up. And still it is a problem, but you are seeing in the pie chart, the brown color is the DMV 1639. Okay, it's a respiratory virus, and it ends up in the lungs. It causing problems, lesions in the upper respiratory tract. And in the lung, the macrophages are infected. That we proved somewhere around 2016-17. At the same time, there was a paper came from Belgium, and it proved IBV replicate in the monocytes. So the virus, if it is in the monocytes and the macrophages, through the lymph, through the blood, it can spread to the rest of the body. It can end up in various body systems and replicate and cause problems. The virus is excreted through the oropharyngeal route as well as through the cloacal route. The SARS coronavirus vaccine was entirely based on the S1. And that may be one reason, because it's an mRNA vaccine, that may be the one reason why the SARS coronavirus is replicating in the vaccinated individuals. When I looked at the avian corona, I can see that the neutralizing epitopes are not only present in the S1, it's also present in the N and the M proteins. The genome is around 27 kb, and major part of the genome encode for the non-structural proteins. And the structural proteins are encoded towards the 3' end of the genome. in 2017, 18, 19, we analyzed a lot of diagnostic samples. The virus can be isolated from the fecal tonsils, kidney, trachea, tracheal swabs, even pleural swabs. What we did, we isolated the virus, we whole genome sequence, and when we analyzed it, we found it all fit into the G117 lineage. That is where the DNA 1639 belongs. When we align our isolates, the whole genome of our isolates with the Connecticut vaccine and also the 491 vaccine, we saw a lot of similarities. You can see that when it is different, the vertical line is black color. The gray lines indicate the similarities. And if you compare the Connecticut vaccine and the DMV 1639, you can see there are a lot of similarities. This is another way of showing it, the simpler. And you can see that the genome starting from around 12,000 base pairs to 20,000 is almost 100% matching to the Connecticut vaccine strain. Between 4,000 to 12,000, it's almost similar to the 491 vaccine. So what does it indicate is the DMV 1639 present in Canada is a recombinant virus. And it has picked up segments, major parts of the genome from the two vaccines. And also you can see that some of the segments towards the 3' end, which is coding for the N, also coming from the Connecticut vaccine. So in this point, I need to tell in Canada, there are vaccines, but it's mainly the mass and the con, only 2 serotypes, but multiple serotypes of field virus is present. And we know the coronavirus vaccines are serotype specific, genotype specific. But when you combine several vaccination strategies, you can induce a protective immune response. So we got this virus, first we need to prove whether this is pathogenic. So what we did, we infected the birds at the peak of lay. And you can see that the red line indicates the infected egg production. There's a drop of nearly 40% drop in the egg production. When you open up those birds, you can see that the ovary is regressed and the oviduct is atrophied. In your left, what you are seeing is a control with nice ovary, with a lot of developing follicles and the well-developed oviduct. In general, avian coronavirus induce egg quality issues. You can see that the egg shell can be decolored. There can be wrinkles. And if you crack an egg from an IBV infected bird, you can see that the albumin is thinner, watery. With the vaccination, you can keep the poor egg quality issues to about 5%, but without a vaccine, the poor quality eggs can increase to about 25 to 30%. We looked at the oviduct at several locations, magnum isthmus, shell gland, and the ovary, and we saw the viral replication. We compared the DMV 1639 with the mass for the egg production drop. And we saw when compared to the mass, DMV 1639 decreased the egg production significantly. And also you can see that the viral shedding is higher in the DMV 1639, particularly through the cloacal route. We looked at several tissues for the presence of virus. We looked at the respiratory tract, kidney, and the reproductive tract, as well as we picked up a couple of immune organs like spleen and the cecal tonsils. You can see that in the cecal tonsils, both the mass and the DMV 1639 had higher viral loads, and the spleen comparatively lower. And also you can see the viral replication, which is assessed by the immunohistochemistry. It is high in the cecal tonsils and lower or not present in the spleen. This is more severe in the DMV 1639 infected birds. We thought to find some correlates of IBV persistent in the cecal tonsils, and we found the T regulatory cells are higher when compared to the spleen in the cecal tonsils. And the CD8 positive T cells are comparatively lower in the cecal tonsils when compared to the spleen. So that nicely fit into the story of higher viral persistence in the cecal tonsils. Okay, we looked at the lesions, and you can see that the lesions are higher significantly in the reproductive tract, kidney, and lungs in the DMV 1639 infected birds. We looked at the rate of apoptosis of the cells in these tissues, except in the respiratory tract, in the kidney, ovary, and the reproductive tract. The apoptosis rate is higher in the DMV 1639 infected birds. So what will happen if we infect the birds at the age of 1 or between the 1 and 14? And it can induce a cystic oviduct lesion. When the oviduct becomes cyst, what is going to happen is these birds are healthy. They are going to eat and drink like a normal bird, and they pretend that they are laying eggs. That's what you are seeing in the middle. They are pretending, but there's no egg production. When you open up these birds, you are going to see the cyst, and also you are going to see the egg yolk in the abdominal cavity. What is going to happen is when the egg is released into the infundibulum, it rolls down, and when it hits the cyst, it cannot pass, then it will back up. When it back up, it go and fall into the abdominal cavity through the infundibulum. So these birds are not going to recover. What we saw was 46% cystic oviduct. So in a flock of 100 birds, 46 birds are not going to produce a single egg. So in Canada, the response is just half the flock. Kill all the birds and shut it and restock it, right? So this is a major loss when compared to the infection in the peak of lay. That is transient and it can bounce back within 10, 12 days. Okay, the little bit of immune suppression, we saw that the virus replicate in the bursa of fabricius. That is the B cell organ, what you are seeing in the immunohistochemistry pictures. And when we look at the histology, what we are seeing is there's a depletion of B cells. In the control, you can see the B cells are packed and nicely arranged. And around starting from day 3, 7, and 10, you can see the depletion of the B cells in the basal follicles and formation of cysts. And that's the indication that the B cells are lost. And we thought to see whether there's a direct replication of the virus in the B cells. Through in situ hybridation, we found the virus is in the B cells. And also we counted the B cell numbers, and there's a loss of around 20% B cells from these DMV 1639 infected birds. So if I summarize quickly about the pathogenesis, when you infect at the peak of the lay, there's a 40 to 60% reduction in the egg production, but it can bounce back within 10, 12 days. When you infect the birds around day 1 to 14, it's going to induce the cystic oviduct that can lead to the forced layer syndrome, and those entire flock will be hulled if it is a drop of egg production by about 20%. And also the IBV can infect the monocyte macrophages as well as the B cells. The functional consequences of the viral replication need to be assessed. So for IBV control in Canada, you need to vaccinate the flocks multiple times because the IBV vaccine-induced immunity is only about 4 to 5 weeks is strong. After that, it is going to decline. And also it is serotype specific. Because of that, what we do is we combine the mask and also the con and vaccinate with the live attenuated vaccine about 3 times, starting for the 2 weeks. First 2 weeks, we are not worried because there will be maternal antibody that is enough to protect it. Then, just before the lay, we induce the inactivated vaccine to boost the antibody and T cell response. So we did a kind of a breeder study. We vaccinated the layers, then we wait until they lay the eggs with a control, and then we collected the vaccinated eggs, the eggs coming from the vaccinated birds and the eggs coming from the unvaccinated birds, and we hatched them, got their progeny, and used for the experiment. And also the vaccinated hen, we used to challenge it to see the protection. So you can see that the antibody response after 3 live attenuated vaccine, it almost doubled with the inactivated vaccine. And also we need to remember that titer is really important because only 30% of that antibody or the immune response is going to be transferred to the progeny, only 30%. So when we challenge the hens, which are vaccinated as well as unvaccinated, you can see that the unvaccinated birds reduce the egg production by about 40%. Yes, almost done. Yes, almost, and all other vaccines are kind of inducing the protection. And also, the maternal antibodies are good enough to protect from the cystic oviduct lesions and also the immune suppression. With that, I'll quickly summarize these outcome. One, the field virus DMV 1639 is a recombinant, and it can induce the egg production drops, cystic oviduct. Because it is a recombinant vaccine, sorry, the recombinant virus, the vaccines which composed of the mass and fun are adequate to induce the protection. And again, I'd like to thank the Department of Poultry Science of this university for the opportunity to come over here, my collaborations, my students, and also the funding agency. Actually, the work I summarized are from work of 3 PhD students and 4 MSc students. One of them is Farooq and is here with us today. Thank you very much.